Scientists just created the world's first AI-designed molecular assassins that hunt down and destroy cancer cells with surgical precision. These aren't drugs or treatments. They're custom-built protein missiles that turn your own immune cells into cancer-seeking guided weapons. And they're about to change everything we know about fighting cancer. But here's what makes this breakthrough truly extraordinary. What used to take years of painstaking research can now be accomplished in just weeks, potentially bringing personalized cancer treatments to millions of patients who are running out of time. Welcome back to AI Handbook channel, where I expose the revolutionary changes happening in artificial intelligence every single day. By the end of this video, you will understand how AI just solved one of medicine's greatest challenges, why these molecular missiles might be the key to defeating cancer forever, and how close we really are to living in a world where cancer becomes a manageable disease rather than a death sentence. On July 24, 2025, the journal Science published what might be the most important medical breakthrough of our generation. A team from the Technical University of Denmark and Scripps Research Institute didn't just develop a new cancer treatment. They built an AI platform that designs molecular assassins on demand. Here's how revolutionary this is. Traditional cancer immunotherapy involves finding natural T-cell receptors that can recognize and attack cancer cells. It's like hunting through millions of keys to find the one that unlocks a specific door. This process takes months or years, costs millions of dollars, and often fails completely. But these scientists flipped the script. Instead of hunting for existing keys, they taught AI to manufacture perfect keys in a computer. Their platform uses 3 AI models working in perfect harmony. RF diffusion analyzes the cancer target structure. A second AI model generates thousands of protein designs that could bind to that target, and a third AI model evaluates and refines the results. The result? Custom-designed proteins called mini-binders that attach to T-cells and guide them to cancer with unprecedented precision. When researchers tested these AI-designed proteins on melanoma cells, the results were stunning. The engineered T-cells, which they named IMPAC-T cells, didn't just find cancer, they hunted it down and destroyed it with the efficiency of guided missiles. But how is this different from existing cancer treatments, and why should this give hope to millions of cancer patients around the world? To understand why IMPAC-T cells are revolutionary, you need to understand the fundamental problem with current cancer treatments. Cancer is essentially a stealth disease. Cancer cells are masters of disguise that can hide from your immune system by looking almost identical to healthy cells. Traditional chemotherapy is like carpet bombing. It attacks everything and hopes to hit the cancer harder than it hits healthy tissue. Radiation therapy is like using a sledgehammer when you need a scalpel. Even newer immunotherapies often struggle because they can't tell friend from foe with perfect accuracy. IMPAC-T cells solve this problem by giving your immune system superhuman vision. The AI-designed mini-binders act like molecular GPS systems, programming T cells to recognize specific protein fragments that only appear on cancer cells. It's like giving your immune system X-ray vision that can see through cancer's disguise. In laboratory experiments, IMPAC-T cells engineered with AI-designed proteins could rapidly kill melanoma cells and prevent cancer growth. But here's the most exciting part. The researchers didn't just test this on one type of cancer, they successfully designed mini-binders for NY-ESO1, a protein found across many different cancer types, and they also created personalized binders for a metastatic melanoma patient's specific tumor. This means we're not just talking about a single new treatment. We're talking about a platform that could potentially create personalized cancer treatments for any patient, targeting their specific tumor's unique fingerprint. Dr. Christopher Haram Johansson, the study's co-author, captured the excitement perfectly. It was incredibly exciting to take these mini-binders, which were created entirely on a computer, and see them work so effectively in the laboratory.
But creating effective cancer killers is only half the battle. How do you ensure these molecular assassins don't attack healthy tissue?
One of the most brilliant aspects of this AI platform is what the researchers call virtual safety screening. Before any AI-designed protein ever touches a living cell, it undergoes rigorous computer testing to predict whether it might accidentally attack healthy tissue. This is crucial because the history of medicine is filled with treatments that worked in the lab but caused devastating side effects in patients. The AI platform addresses this by screening thousands of potential mini-binder designs against databases of healthy human proteins, effectively running millions of safety tests in silico before moving to laboratory experiments. Professor Saine Riker-Hadrup, co-author of the study, emphasized this point. Precision in cancer treatment is crucial. By predicting and ruling out cross-reactions already in the design phase, we were able to reduce the risk associated with the design proteins and increase the likelihood of designing a safe and effective therapy. This virtual safety screening represents a fundamental shift in how we develop medical treatments. Instead of discovering dangerous side effects during expensive clinical trials, we can identify and eliminate them during the design phase. It's like having a crystal ball that shows you the future consequences of your medical decisions. The platform's safety features are so sophisticated that they can distinguish between proteins that look almost identical but have vastly different functions. This level of precision means we could potentially develop cancer treatments that are both incredibly effective and remarkably safe. But perhaps the most game-changing aspect of this technology isn't just what it can do, it's how fast it can do it. Here's where this story becomes truly extraordinary. The entire process of designing, testing, and validating a new cancer-targeting protein now takes just 4 to 6 weeks instead of multiple years. Let that sink in for a moment. A cancer patient diagnosed today could potentially have a personalized treatment designed, tested, and ready for production in less time than it currently takes to schedule most medical procedures. The speed improvement isn't just convenient, it's literally life-saving. Cancer doesn't wait for slow research cycles. Every month of delay means more patients lose their battle with the disease. This AI platform compresses decades of traditional drug development into weeks of computational work. Dr. Timothy Jenkins, the study's lead author, explained the timeline. It takes as little as a day or two to come up with promising designs, and just a few weeks to test them in the lab. That's faster than current methods, which can involve hunting through people's cells to pick out proteins naturally able to lock onto specific cancer targets. The platform's speed comes from its ability to design proteins entirely in computers, rather than having to find them in nature. It's like the difference between growing your own food versus ordering delivery. Both get you fed, but one is infinitely faster when you're hungry. This speed advantage becomes even more critical when you consider personalized medicine. Traditional drug development assumes one treatment fits all patients with the same cancer type. But every tumor is unique, with its own genetic fingerprint and protein signatures. The AI platform can potentially create custom treatments for each patient's specific cancer profile. But speed and personalization are just the beginning. The implications of this technology extend far beyond individual treatments. This breakthrough didn't happen in isolation. It builds directly on the computational protein design technologies that earned David Baker the 2024 Nobel Prize in Chemistry. Baker's lab at the University of Washington has now become a launching pad for revolutionary medical technologies. In fact, the commercial potential of this AI platform is so promising that the University of Washington researchers are planning to launch a company to bring these treatments to market. This isn't just academic research gathering dust in journals. It's technology that's actively moving toward real-world application. Since 2014, Baker's Institute for Protein Design has spun off 10 startups, and Baker himself has co-founded 21 tech companies. When the Royal Swedish Academy of Sciences awarded him the Nobel Prize, they specifically noted that his research group has produced one imaginative protein creation after another, including proteins that can be used as pharmaceuticals, vaccines, nanomaterials, and tiny sensors. The IMPAC-T cell platform represents the next evolution of this Nobel Prize-winning work. Julia Bonzani, a co-lead author and Baker Lab graduate student,
expressed the humanitarian drive behind the research. I'm hopeful that this will lead to new therapies that are more accessible to patients around the world who do not benefit from current state-of-the-art cancer treatments. This focus on accessibility is crucial. Current personalized cancer treatments can cost hundreds of thousands of dollars and are only available at the world's most advanced medical centers. An AI platform that can design treatments computationally could potentially democratize personalized medicine, making cutting-edge cancer therapy available to patients regardless of their location or economic status. But the most exciting aspect of this technology might be how it could transform cancer from a death sentence into a manageable chronic condition.
Dr. Jenkins estimates that it will take up to 5 years before this technology is ready for initial clinical trials in humans. In the world of medical research, 5 years is lightning speed. Most cancer treatments take 10 to 15 years to move from laboratory to patient. Once approved, the treatment process will be remarkably similar to current CAR T-cell therapy, which is already used to treat lymphoma and leukemia. Patients will have blood drawn in a simple procedure similar to a routine blood test. Their immune cells will then be extracted and genetically modified in the laboratory to carry the AI-designed mini-binders. The modified cells will be infused back into the patient, where they'll circulate through the bloodstream as molecular assassins, seeking out and destroying cancer cells with unprecedented precision. The entire process, from blood draw to treatment, could potentially be completed in weeks rather than months. But here's what makes this particularly exciting. Unlike traditional drugs that work for some patients but not others, the AI platform can potentially design custom treatments for each individual patient's unique cancer signature. It's like having a tailor-made suit, but for your immune system. The researchers have already demonstrated this personalized approach by successfully creating mini-binders for a metastatic melanoma patient's specific tumor markers. This proof of concept shows that the platform can adapt to any cancer target, opening the door for treatments that are literally designed for each individual patient. The global implications are staggering. If this technology proves successful in clinical trials, we could be looking at a future where cancer treatment is fast, precise, personalized, and accessible to patients worldwide. We're standing at the threshold of a new era in cancer treatment. For the first time in human history, we have the ability to design molecular weapons that can distinguish between healthy cells and cancer cells with perfect precision, and we can do it faster than the cancer can evolve. This isn't just an incremental improvement in existing treatments. It's a fundamental paradigm shift. We're moving from a world where cancer treatment involves educated guessing and hoping for the best, to a world where treatment involves precise engineering and designing for success. The AI platform developed by these researchers represents something unprecedented: the ability to turn cancer's greatest strength against itself. Cancer's ability to mutate and evolve has always made it difficult to treat. But when you can design new molecular assassins in weeks instead of years, evolution becomes an advantage for the treatment, not the disease. Dr. Baker captured this transformation perfectly. Detecting unhealthy cells is one of the main jobs of the immune system, but it doesn't always notice the subtle signs of cancer or viral infection. In this study, we show that computer-designed proteins can help human immune cells spot the right targets and function more effectively. The implications extend beyond cancer. The same AI platform that designs cancer-fighting proteins could potentially be adapted to target viral infections, autoimmune diseases, and other conditions where precise immune targeting is crucial. The researchers have already demonstrated this versatility by creating proteins that target both cancer cells and virus-infected cells, showing that the platform's applications could be virtually limitless. We're witnessing the birth of a new field: computational immunotherapy. Instead of relying on chance discoveries or lengthy trial and error processes, we can now engineer immune responses with the precision of software development and the speed of artificial intelligence. The molecular assassins are here. They're designed by AI, guided by precision, and powered by hope.
The question isn't whether they'll change cancer treatment, it's how quickly we can get them to the patients who need them most. The revolution in cancer treatment isn't coming, it's already here, designed one protein at a time by artificial intelligence that never sleeps, never gives up, and never stops hunting for new ways to save human lives.